Hi everyone, welcome to lesson 4.4 on non-Mendelian inheritance. As always, as we go through this lesson, please make sure to fill in your guided notes and keep up with your vocabulary. So far, throughout Unit 4, we've been discussing Mendelian inheritance, and that shows dominant and recessive alleles of one or multiple genes. For example, yellow versus green seeds, round versus wrinkled seeds in pea plants. Really, whenever you see Mendelian inheritance, we're kind of thinking like simple, dominant, recessive type genes, okay? Non-Mendelian inheritance, what we're going to discuss today, are patterns that are not determined by simple, dominant, or recessive alleles, and they can involve one or multiple genes. So when you look at people and animals, think to yourself, do you think our genes are determined by simple, dominant, and recessive traits? If you thought, well, no, probably not, you're correct. Most of our genes are not just simple, dominant, recessive, okay? As a matter of fact, we're going to look at a couple different inheritance patterns that will involve us today. So we will discuss incomplete dominance, codominance, multiple alleles, sex-linked inheritance, polygenic traits, and genes and environmental conditions. So again, this is a lot of information. On your guided notes, make sure to really write out and show your work so that way you can recall and understand what you did in the next class. So our first scenario, we're going to look at incomplete dominance. So some alleles are neither dominant or recessive. So here's your definition, and incomplete dominance is cases in which one allele is not completely dominant over another. A classic example of incomplete dominance are 4 o'clock plants and snapdragons. And here, okay, here's a good kind of Punnett square, but red flowers here and white flowers, when mixed, can make pink flowers. So this is kind of showing that one allele is not completely dominant over the other. Now, how do we indicate incomplete dominance patterns? Well, here, this is a good way. So one thing that you can do to show that it is an incomplete dominance pattern, you can have a C, okay, for color, and then put like double R for red. Same thing, CRCW would stand for pink, and CWCW would stand for white. So really, this is kind of up to you on how you want to write out your genotypes for incomplete dominance. I will be using this one as we go through this lesson because I think having that C for color base helps me distinguish incomplete dominance versus something like simple dominant and recessive a little bit better. So here's our first practice problem. So the snapdragon is a plant that shows incomplete dominance inheritance pattern. Homozygous white flowered plants have the genotype CWCW, and homozygous red flowered plants have CRCR. Pink flowers will be CRCW, or that mix of those two. So first we're going to show a cross between a white flowered plant and a red flowered plant. So step one is the same as our other Punnett squares. We have to determine what the parents are. Okay, so our parent 1 genotype, CWCW for white, parent 2, CRCR for red. Okay, set up your Punnett square and go ahead and cross your parents. Parent 1, parent 2, and we should get something that looks like this. So here's your red parent, here's your white parent, we cross them, and we end up getting all of them CRCW. Again, if you're confused, why are we using the C's? This is what I like to use to make sure that I know it is an incomplete dominance pattern, or really a non-Mendelian inheritance pattern, rather than just simple dominant and recessive. So here we have all CRCW offspring. What did that mean? Well, here, that would be pink. So your expected offspring from this cross are 100% pink. All right, problem 2. So I want you to now try this one on your own. So pause here, try it out, and then come back once you are finished. All right, so this one we are looking at a pink flowered plant, so we should have CRCW for pink and red flowered, CRCR. Set up your Punnett square, put up parent 1 and 2. Okay, and let's go ahead and look at the offspring. So your pink flowered plant is here, red here. We do this cross and we should get 50% would look red, right? Expected to have 50% offspring look red here. And we'd expect 50% of the offspring to look pink. All right, so that was our second incomplete dominance example problem. All right, the next pattern that we're going to talk about is codominance. Now, codominance is when cases in which phenotypes produced by both alleles are clearly expressed. Now, keep in mind it's really easy to get this one confused with or mixed up with incomplete dominance. Remember, here's your key word. Both alleles are clearly expressed. right? So that's kind of the key word for that definition. Human blood types demonstrate codominance. So, for example, type AB blood has both A and B clearly expressed. And in some chickens, black feathers are codominant for white feathers, and they produce what we call speckled or erminet chickens. So, this would be an example of codominance because you can clearly see both white and black. Okay, so here's our first codominance example problem. Chickens can exhibit a codominant inheritance pattern. White chickens, we're going to use that same C for the color base, are CWCW, and homozygous black feathered chicken, CBCB. Erminet feathered, which again was that mix, okay, of white and black, both clearly expressed, are CBCW. So we're going to show a cross between an erminet chicken and a black chicken. So first off, parent 1 genotype would be CBCW. Parent 2, CBCB for black. Okay, set up your Punnett square. Go ahead and try out that cross on your own. And we should end up getting something like this, okay? We would end up having our 2 black chickens, and then we would also have 2 speckled or erminet chickens, okay? Both clearly exhibiting the black and white, again, because this is codominance. All right. Next pattern, okay, or next mode of non-Mendelian inheritance, multiple alleles. So multiple alleles are a gene that has more than 2 alleles. So this is a pretty easy definition to remember. So Mendel studied 2 alleles of his pea plant, round seeds or wrinkled seeds, but real populations usually have multiple alleles. So for example, we have 3 alleles for human blood type, A, B, and O. Rabbit coat color is also determined by a single gene with 4 alleles that can produce 4 coat colors. So here are your examples here. Black, chinchilla, Himalayan, or albino. These are those 4 multiple alleles. So human blood type are going to be our example problems for multiple alleles. Red blood cells carry antigens which trigger an immune response. There are 2 types of antigens, A and B. People with type A blood have the antigen A, type B have the B antigen, and AB have both. Type O has neither. Both A antigens and B antigens are clearly expressed and are written as follows. I is going to be that base, I big A and I big B, while having no antigens is written as a little I. So here's kind of a good table breaking down all the possible blood types and the combinations. If this table is confusing, then I went ahead and basically wrote it out right here. So blood type A could have 2 big A's, or it could have the A antigen with the O. Okay, same thing. Type B could have 2 big B's, or IB with a little I. Okay, type AB only has 1 possible combination, IAIB, and type O has the 2 lowercase i's because, again, it does not have any antigens. If you want more information on this, there's a really good video linked back in Blend on blood type. It's optional, but I recommend watching it because it's a good video. Okay, so here are our blood type practice questions. Go ahead and we can pause here. Try to answer the first one on your own, and then we will go through these 2 together. All right, number 1. So since blood type has 2 or more clearly expressed alleles, which is codominance in the forms of A and B, blood type also is a gene with more than 2 alleles because we have A, B, and O. So that is how we get both codominance and multiple alleles. Okay, number 2. So now it's going to be where we do Punnett squares on blood type. Could a person with type AB blood have parents with type O blood? Perform a Punnett square and explain. So the first thing is we're going to start this problem the exact same way that we have been. We have to find out what our parents are. So both parents have type O, so parent 1, genotype, little I. Same thing for parent 2, okay? Now go ahead and set up your Punnett square, and we get 100% is going to look like type O. So no, it is not possible for a person with AB blood to have parents with type O blood. Okay, number 3, could a person with type O blood have a parent with A and a parent with B blood? So perform a Punnett square and explain. Now before we do this problem, there are 2 possible genotypes for type A and type B, okay? Now, as you're thinking to yourself and setting up your Punnett square, it's just asking, is it possible? So you have to look at the combination that could possibly produce type O. So instead of using these two, okay, these two possible genotypes, we will be using these two. So parent 1 genotype would be capital A with that little I. Parent 2, same thing, capital B with the little I, because they need to have that recessive O antigen or recessive O allele in order for this to happen. Again, if they did not, there is no possible way that they would have type O. So we set up our Punnett square, perform your cross, and yes, it is possible, right? So we end up getting, okay, 25% are AB, okay, expected to have 25% AB, okay, we would have 25% type A, 25% type B, and last one, 25% type O. So yes, it's possible, but again, only possible if both of the parents have that recessive I allele. If they do not, then no, we could not get type O from this. Okay, now we're going to look at our next mode of inheritance, and that is sex-linked. So sex-linked inheritance occurs when there is a gene for a trait that is found on the X or Y chromosomes. Okay, the male sex chromosomes are XY, female are XX. Genes on the Y chromosomes will be found only in males, again, because only males have the Y chromosome. Recessive genes found on the X chromosome will be more likely in males than females. Okay, and we're going to look at a couple practice problems of why that is. So, Punnett squares for sex-linked traits look a little bit different. The Y chromosome is only found in males, and for our practice problems, it's never going to carry alleles, okay? Now, what does that mean? Whenever we set up a Punnett square, we will never have a little letter next to the Y, okay? Because we're not going to have to do that type of inheritance pattern in this class. The X chromosome is found in males and females and can have alleles. Males can either have a trait on the X chromosome or they don't have it. Females can have the trait or be carriers for that trait. Now, what does that mean? Okay, so here is a classic example of a sex-linked trait, okay, hemophilia. So a normal female would have 2 copies of this capital allele, okay, because this is, in this case, a recessive disorder, all right? A normal female would also be heterozygous, right? Because being normal is dominant, if she's heterozygous, she is still normal. She does not have hemophilia. However, she is considered a carrier. And what that means is that she can just pass this little H along to a child. And then the only way a female can have hemophilia if she has 2 copies of the recessive allele here, all right? For a male, he has to have 1 copy, okay, of that capital letter, or that 1 copy of that dominant trait to be normal. And if he has 1 copy, okay, of the lowercase letter, or in this case, the recessive trait, he will have hemophilia. So this is what we mean by you do not put any alleles, okay, on the Y chromosome. We don't have to worry about that. We're only going to ever put alleles in this class on the X chromosome. So here is an example, okay? This was not on your notes, so just kind of watch this one. This would be considered a normal male, okay? This would be considered a normal but carrier female. If we cross these two, what we end up getting is 1, okay, normal female. This female is also normal, but she is a carrier. We also end up getting 1 normal male and 1 male that does have hemophilia. All right, so this is going to be kind of how we set up our practice problems. All right, so first example, hemophilia is a sex-linked recessive trait. Show a cross between a normal non-carrier female and a male with hemophilia. So I went ahead and put this key up here so you can go ahead and look at that. But parent 1 is a non-carrier normal female, okay, so here. And a male with hemophilia would be this. So parent 1, female, parent 2, male. Okay, now we set up your Punnett square. Put parent 1, parent 2, and we're going to go ahead and do your cross. Now, this first one, okay, remember your X's go together, and then X will go before the Y. So here we have, okay, both of the female offspring, okay, or if you're looking at it like this, 100% of the female offspring are carriers for hemophilia, or they are normal, right? They do not have hemophilia, they could just pass it along, okay, while 100% of the male offspring are normal, okay, or both of the males are normal. So you notice that these ratios are a little bit different. We're not saying like 50% of the females, right? Because we're looking at both females, so now it's out of that 100%. All right, problem 2, okay? So now that we've gone through that example, you can pause here and try this one on your own. Okay, so this one, we are showing a cross between a carrier female and a normal male. So our carrier female, okay, again, has to have that recessive allele, okay, because it's a sex-linked recessive trait. So she is normal, but a carrier. Our male is normal, okay, so he has the capital allele. Go ahead and put parent 1, parent 2, perform that cross, and we should get here, okay? Now we have 50% of the female offspring, right? Because we're looking at females and males, okay? 50% of the female offspring is completely normal. 50% of the female offspring would be considered a carrier, okay? Although still normal, she would be a carrier. 50% of the male offspring would be normal, and 50% of the male offspring would be would have hemophilia, right? Males cannot be carriers, okay? So that's kind of how we got that breakdown here. All right, check for understanding. Okay, so here are 3 practice problems. I want you to try to pause here, try these 3 on your own, and then we will go through them together. Okay, so number 1, all right, read through the problem, and here's what we need to get first. Think first off, okay, what inheritance pattern are we looking at? And it says it in the problem. They are co-dominant, okay, so that is our first thought. Second thing, we need to know what both parents are. So here it says determine the phenotypes of the offspring between 2 heterozygous griffins. So both parents are heterozygous, and I'm going to put for this, okay, B for brown and G for gray. All right, so both parents would have this genotype. Okay, now step 2, we set up your Punnett square. All right, both parents are heterozygous, therefore they look the same. Go ahead and do this cross, and we get 25% of the offspring would be brown. Okay, 50% would have both brown and gray. Again, because it is co-dominant, okay, both are clearly exhibited. So 50% brown and gray, and 25% would be gray. Okay, number 2. So we should have read through that problem. All right, now we're going to kind of break this down a little bit more. So first off, what inheritance pattern are we looking at? We are looking at a sex-linked inheritance pattern, okay, because it's telling you gene on the X chromosome, right? Second thing, homozygous recessive, it tells us means that it has small claws. So the male is small clawed, the female is heterozygous, or she is a carrier for small claws, but she still appears to have large claws. So let's go ahead, and now that you have this information, now you can try this out on your own. All right, so, okay, your genotypes, I'm going to use H's again just because that's what we have been using so far. So again, because your male is small clawed, he is recessive. You would have to have a lowercase letter. Again, don't put any letters next to the Y. And the female, okay, is a carrier, so you would have 1 capital and 1 lowercase letter. All right, so we have your male, okay, small clawed, female, normal, but a carrier. And we go ahead and do this cross. And here is what we would get. We would end up getting 50% of our female, again, we're going out of the females, okay, 50% of the female offspring are large clawed carriers, 50% would have small claws for the females, okay, males expected 50% to have large claws and 50% to have small claws. Okay, last problem on the check for understanding. Okay, first off, the inheritance pattern is incomplete dominance. Okay, it tells us that in the problem. Second thing, both parents are heterozygous. So for our genotype, since it told us red and yellow, I'm going to use CRCY, okay, to show that that is orange. So we do our cross, okay, both of our heterozygous parents, and here would be our offspring. So we would get 25% red, 50% would be orange, okay, again, because it's incomplete, okay, they are both kind of blended together, and we'd have 25% that are yellow. All right, last couple things. So, polygenic traits are traits controlled by two or more genes, and they often show a wide range of phenotypes. So, some common examples of polygenic traits are human height and pigmentation of skin. And remember, many traits that whenever we look at things like humans, animals, many traits exist on a continuum rather than in discrete forms. And by discrete forms, we mean simple, dominant, and recessive, right? If brown eyes were just simple dominance over blue eyes, there wouldn't be such a wide variety of eye colors. Now, next thing, this was mentioned back in Unit 3, okay, but this is looking at a different type of inheritance, okay, maternal inheritance due to mitochondrial and chloroplast DNA. If you remember, okay, mitochondria is the organelle that converts chemical energy stored in food into compounds that the cell can use. This was also the site of cellular respiration, which is why we called it the powerhouse of the cell. In humans, all or nearly all of our mitochondria come from the cytoplasm of the ovum or egg cell. Essentially, we get all of our mitochondria from our moms. Now, this one, okay, helped us support endosymbiotic theory, and two, something that you need to understand is that you only inherit mitochondria from your mother. Therefore, all offspring will have identical mitochondrial DNA to their mother. If you want to read more on this, there's a really good article you can search and read that. Now, last one, okay, genes in the environment. So, environmental conditions can affect gene expression. The phenotype is only partially determined by the genotype whenever, again, environmental conditions can come into this. So, the wing color of the western white butterfly is variable depending on the temperature, and that allows the butterfly to optimize wing temperature to accommodate flight. Another example of genes in the environment are hydrangeas. Here, they can change color based on soil pH. So there's a couple other examples of this, and if you can find a couple other examples of genes in the environment, please do so and write those down on your notes. All right, so quick check for understanding just based on those last two things. Pause here and try this out. All right, so first one. If she has 4 children, how many of them would you expect to inherit this trait that was in her mitochondrial DNA? So again, all 4 children, or 100% of them, will inherit her mitochondrial DNA, just because that is the way in which we inherit mitochondrial DNA. Number 2A, okay, we just went over this example. This would be the environmental influence on genes, okay, and part B, human weight doesn't fit into two phenotypic categories, so we would call that polygenic, right? It's influenced by many genes and it's more on a continuum. All right, we have reached your exit ticket. I want you to pause here and try out both of these on your own. Okay, so number 1. Again, we already did the snapdragon example, so pink flowered and white flowered plant. Okay, parent 1 would be pink, parent 2 white. We go ahead and do that cross, and 50% would be expected to be pink, okay, and 50% would be expected to be white. Last one, okay, possible blood types of offspring from 2 parents who are both AB blood type. So parent 1 genotype, parent 2 genotype, both AB. We perform this cross and we get 25% expected to be type A, 25% expected to be type B, and 50%, or these two, expected to be type AB. That is the end of lesson 4.4. Thank you for watching. Please again make sure to have written down your examples, show your work, and reach out with any questions.